This data table tracks the epidemiological outcomes of the MESA cohort, a longitudinal study monitoring nearly 6,800 healthy adults over a 16.9-year period. The analysis identified a molecule correlating to a 12% increased hazard ratio for all-cause mortality and a 44% increased risk for fatal kidney failure. Expanding the interquintile range reveals a stark dose-dependent relationship. As plasma concentrations climb, renal failure risk compounds. These elevated concentrations remain a significant, independent predictor of death. Even after researchers adjust for baseline kidney function, blood glucose, and pre-existing comorbidities. The molecule is trimethylamine N-oxide, or TMAO. It is highly water-soluble, distinguished by a polar nitrogen-oxygen bond, and is not synthesized by human tissue in isolation. TMAO is a lethal, active architect of systemic cellular senescence and cardiometabolic decay. To understand the origin of TMAO, we must observe the human body through a meta-organismal lens, a framework where the host and its gut microbiome function together as a single, highly integrated metabolic engine. Understanding this molecule requires moving away from the reductionist view that human metabolism happens in isolation. The systemic accumulation of TMAO relies on a three-part biochemical cascade: the dietary ingestion of quaternary amines, enzymatic cleavage by gut bacteria, and subsequent oxidation by the human liver. The primary exogenous substrates driving this pathway are L-carnitine and phosphatidylcholine. These precursors are overwhelmingly concentrated in animal-sourced foods, particularly red meat, organ meats, and egg yolks. Because TMAO synthesis requires both a specific dietary payload and a specific microbial enterotype, the entire pathological cascade is highly vulnerable to targeted clinical intervention. This vertical schematic maps TMAO synthesis, beginning when dietary precursors like L-carnitine and choline arrive in the lower gastrointestinal tract. Within the lumen, Firmicutes bacterial strains deploy the CUT-C/D gene cluster. These microbes cleave the carbon-nitrogen bonds of dietary substrates, producing trimethylamine, or TMA. This toxic intermediary gas diffuses across the intestinal mucosa into portal circulation. Arriving at the liver, the host enzyme FMO3 oxidizes the TMA into stable TMAO, which is immediately released into the systemic bloodstream. There is one major clinical exception to this meta-organismal pathway: deep-sea marine seafood. Saltwater fish naturally synthesize massive concentrations of pre-formed TMAO to stabilize their proteins against deep ocean pressure. When humans consume these marine organisms, the intact TMAO bypasses microbial cleavage and absorbs directly into the bloodstream. Outside of saltwater fish, standard Western diets reliant on terrestrial animal proteins construct an efficient microbial hepatic assembly line, ensuring a perpetual flood of atherogenic TMAO. Looking at this arterial cross-section alongside the hepatic mechanism isolates how TMAO drives atherosclerotic plaque. At the endothelial wall, TMAO upregulates CD36 scavenger receptors on local macrophages. This triggers hyperactive uptake of oxidized LDL, transforming macrophages into lipid-laden foam cells. Concurrently in the liver, TMAO suppresses the CYP7A1 enzyme, paralyzing reverse cholesterol transport and trapping excess cholesterol within peripheral tissues and arterial walls. Moving to acute thrombosis risk, this view inside a blood platelet isolates the IP3R channels and SERCA reuptake pump. TMAO acts as a sensitizing agent, directly activating IP3R channels to release massive stores of intracellular calcium into the cytosol. Simultaneously, TMAO inhibits the SERCA pump. Disabling this calcium reuptake mechanism forces a sustained pathological elevation of cellular calcium. This massive calcium load lowers the platelet's activation threshold. The cells become violently hyperreactive, promoting rapid degranulation and exponentially increasing the rate of thrombus formation. TMAO actively primes the vascular network. It builds plaques over time and alters blood biochemistry, making plaques highly vulnerable to catastrophic rupture and ischemic events. This intracellular flowchart demonstrates TMAO operating as a pathological activator of the PI3K-AKT-mTOR signaling cascade. TMAO-induced overactivation of the mTOR network acts as a relentless negative regulator of macroautophagy, suppressing the critical ATG16L1 complex. This failure arrests autophagone construction. Deprived of recycling ability, the cell rapidly fills with toxic protein aggregates and failing organelles. Accumulating dysfunctional mitochondria trigger a massive surge in reactive oxygen species. This initiates the PERK pathway, forcing the cell into chronic endoplasmic reticulum stress. Failing autophagy stops the cell from dividing, but it resists death. It becomes a senescent zombie, secreting inflammatory cytokines that degrade surrounding healthy tissue. By eroding basic cellular survival mechanisms, TMAO drives progressive organ failure, cognitive decline, and diminished health span. Since TMAO generation relies entirely on dietary precursors and a specialized microbiome, the pathway is highly modifiable. This line graph illustrates data from the swap meat crossover trial, providing clinical proof that dietary intervention can quickly extinguish the cascade. When participants transitioned from an animal meat diet to plant-based alternatives, they experienced a rapid collapse in systemic TMAO, dropping by 0.61 log units. Eliminating heavy loads of L-carnitine and animal-derived choline starves the specific TMA-producing bacterial clusters within the lower intestine. Over time, strict adherence to a whole food, plant-based diet alters the host's bacterial enterotype, depressing the populations of firmicutes in favor of beneficial strains. This microbial restructuring essentially eradicates the bacterial strains capable of expressing the CUTC/D lyase enzymes. Long-term vegans often show no elevation in TMAO, even when challenged with a high choline meal. Their microbiome is no longer equipped to execute the conversion. Strategic biochemical machinery driving cardiometabolic decay. While dietary intervention is effective, broad behavioral compliance is notoriously difficult to maintain. For high-risk clinical populations, advanced pharmacological interventions are moving through active development. Early attempts to clear TMAO used broad-spectrum antibiotics, but the strategy failed. Antibiotics indiscriminately destroy beneficial gut flora, and the TMA-producing bacteria rapidly repopulate the gut once the treatment ends. This molecular model illustrates the clinical frontier of precision medicine. Highly specific halomethylcholine derivatives, most notably iodomethylcholine, or IMC. IMC is a mechanism-based inhibitor that mimics choline to penetrate the active site of the microbial enzymes responsible for TMA synthesis, where it irreversibly binds. This permanently jams the enzyme's active site, neutralizing its function without killing the host bacteria or causing disruption to the broader intestinal microbiome. In preclinical models, the continuous oral administration of IMC decouples a high choline diet from downstream TMAO generation, halting the progression of renal tubulointerstitial fibrosis. By mastering the meta-organismal pathway, clinical medicine can move past merely managing downstream lipid metrics. Utilizing precise dietary and pharmacological tools, we can actively neutralize a primary chemical driver of human aging and disease.